After completing basic bacteriology, we will start clinical bacteriology. In clinical bacteriology, we will talk about first gram-positive lab algorithm. In this diagram, we will divide gram-positive bacteria into different groups, and this is very, very important to remember at least. So we have gram-positive bacteria. Gram-positive bacteria that color purple are brief on staining, gram staining. These are three types. One is bacilli, other is cocci, and we have branching filaments. So these are three types: bacilli, cocci, and branching filaments. Bacilli are rod shape, cocci are comma shape, and branching are branching. So these are main three types of bacteria, and then we will discuss further classification of these bacteria. So bacilli are rod shape, cocci are round, are oval, yeah, are comma shape, and branching are branching shape, that they have different branches. First of all, let's talk about bacilli. So bacilli include two types: aerobic and anaerobic. Aerobic are those bacteria that live in the presence of oxygen, and anaerobic are those that are living in the absence of oxygen. So oxygen is toxic to them. Here we have two types which are very important, that include Listeria and Bacillus, Corynebacterium. But the bold one are very, very important, and at least you should remember these. In anaerobes, we have Clostridium, most important, and then we also have QT bacterium, formerly called as a Corynebacterium. So aerobic and anaerobic, these are the two main groups of bacilli. Then if we talk about another small branch, that are branching filaments, also include aerobic and anaerobics. In aerobics, we have nocardia, that is partially or weakly acid fast, we have discussed, and then we have actinomyces, that is not acid stain, not acid fast. So what is acid fast, what is gram positive, what is gram negative, that we have discussed. Then we have cocci. Cocci are round oval shaped bacteria. Then in cocci we do a test that is called as a catalase test. So catalase test is a test that is used to differentiate between Staphylococci and Streptococci. If a bacteria is catalase negative, it is included as in the group of Streptococci. It is Streptococcus that form a chain. As you can see here, this is chain formed by the Streptococci. And if a bacteria is gram catalase positive, then it is called as a Staphylococcus, which form a clusters. Like you can see over here, a cluster of bacteria is formed. So cluster and chains, pairs are chain. Chains are pair formed in the case of Streptococcus, and in the case of Staphylococcus, clusters are formed and they are catalase positive. So remember catalase test. You can write, it is a test used to differentiate Staphylococcus from Streptococcus. If a bacteria is catalase positive, bubbling formation occur, and this is catalase positive, and this bacteria is Staphylococcus. And if a test is negative, that catalase negative, then it will be Streptococci. Then we have Streptococci. If we come towards Streptococci, then in here we do another test, that is coagulase test. So in Staphylococci, in a Staphylococcus, we do another test that is called as a coagulase test. So what is coagulase test? Coagulase is a test that differentiates Staphylococcus aureus from other staphylococcal bacteria. So we have staphylococcal bacteria, Staphylococcus aureus, and if any bacteria that is not Staphylococcus, then it is differentiated by coagulase test. So in this case, a rabid plasma in a tube aid bacteria coagulase, causing fibrin clot to form. So if a bacteria is coagulase positive, fibrin clots formation will occur. If coagulase positive, then clumping will occur, and if coagulase negative, then there will be no clumping. And clumping occur because fibrin clots occur. So in case of Staphylococcus aureus, we have catalase coagulase positive. And if a bacteria is coagulase negative, then we do a test by antibiotic sensitivity, that is no-vicin, which is an antibiotics. We do a test, and this test again gives us two types of bacteria. If a bacteria is sensitive to no-vicin, then it means that it will die because it is no-vicin sensitive, that no-vicin act upon this. So if we say over here is Staphylococcus epidermidis, that is no-vicin sensitive, and Staphylococcus saprophyticus is not sensitive to no-vicin. So you should remember this. So if we here come to our Streptococci, and then we talk about hemolysis. So in this case, we will divide different Streptococci. But first, let's talk about hemolysis. So what is hemolysis? Hemolysis is studied in microbiology. Why we study hemolysis? It is studied in microbiology to identify bacteria, assess their virulence. What is the virulence of that bacteria? First we identify the bacteria, and then we identify or we find the virulence, and guide further diagnostic tests. So hemolysis is done because of this, that what is the bacteria, what is the virulence, and what we'll do with this bacteria. For this we study hemolysis. We have mainly three types of hemolysis. One is alpha hemolysis, beta hemolysis, and we have gamma hemolysis. So in different media bacteria are placed, and that gives us different results. So that is called as a hemolysis. If we talk about alpha hemolysis, then this is a partial hemolysis and it will give green color in media. Then we will do a test of antibiotics, that is optochin sensitivity test, as we are doing in Staphylococci. Here that is no biocin sensitivity. Similarly, here we do biocin, optocin sensitivity, and bile solubility. If bacteria is sensitive, that is positive, so it means optocin sensitivity is present. So if optocin sensitivity is present, it occur in case of a streptococcal pneumoniae, and thus it means that streptococcal pneumoniae will die in the presence of optochin antibiotics. And if this bacteria that is resistant to optochin, then it will revive, it will survive, and it will not die. And these are the example of certain bacteria that will survive in the presence of optochin and bile solubility. Similarly, Streptococcus is bile soluble, while the others are bile insoluble. So this is the alpha hemolysis which occur in this type of bacteria. Then we have beta hemolysis, which is the complete hemolysis, and it will give us a clear color. And again we do a test of antibiotic and PYR status we check. Then that is if sensitivity to bacitracin in a group A, and that is streptococcal pathogens. So in this case these bacteria will die because these are sensitive to bacitracin, and these will not die because these are resistant to bacitracin. So we do bacitracin sensitivity in beta hemolysis. So remember by the word alphabet B. In beta hemolysis we do bacitracin sensitivity, and in alpha you can A and O are related with respect to their sounds. So A and alpha you can remember that optochin sensitivity we do in partial hemolysis. Then here we do now by sensitivity in Staphylococcus in coagulase stage after the coagulase. So these are the different type of bacteria that cause beta hemolysis. In gamma hemolysis there is no hemolysis and there is growth in the bile, and then we check the growth in 6.5% in ACL in PYR status. If positive, then occur in this bacteria, and negative occur in non-Enterococcus, Streptococcus galactolyticus. So this is actually what you should know, that what are the different classes of gram-positive bacteria. Here it is said that important tests are in bold and important pathogens are also in bold. So this is the group of gram-positive bacteria which colored purple or blue in case of gram staining, and we have mainly three branches: bacilli, cocci, and branching filaments. These are various type of tests that we do. In alpha hemolysis we check optochin sensitivity. A and O are similar in sounds. In beta hemolysis we do bacitracin sensitivity, and again one another, that is after the coagulase negative test we do no biocin sensitivity. Now we will discuss each bacteria one by one in the next topics.